about how we produce MUSE cells in the lab. So MUSE cells are found within mesenchymal stem cells. So we have to do the same process of producing the mesenchymal stem cells, which I'll get into here in a minute. But then we have to stress those cells because that's what MUSE stands for. They're multi-lineage stressed enduring stem cells. They basically survive through stress that would kill mesenchymal stem cells. So that's part of the process. Now this goes back to how Dr. Tasawa discovered them in Japan. Apparently they were cultivating mesenchymal stem cells, which the first step is you take the umbilical cord, placenta, whatever tissue you're using, and you start dissecting that. You're then putting it into these cylinders with collagenase and centrifuging, spinning them real fast to separate the cells from the tissue. And after that, you would then normally take them and then centrifuge further to what's called a pellet, then put it in the flask, and then start culturing. Well, they just got to the point where they had centrifuged and something happened. They had to leave the lab and they left, forgot about them, went back the next day. And at this flask with all the collagenase and cells, well, as expected, the cells had died, fallen to the bottom, but there were still some cells that had survived that were at the top. So they're like, oh, wow, that's weird. These should be dead. Let's take a look. What are these? They analyze them with the flow cytometer and they start noticing these have the surface markers, phenotypes of pluripotent stem cells. Right? That's interesting. Research further. Sure enough, they're pluripotent stem cells, which means they can turn into any type of cell in the human body needed. So very interesting. The cool part is they act like mesenchymal stem cells, that they seek out inflammation and damage. They go there, they basically consume or take over the dead cells or injured cells and then turn into that tissue type. So really cool. But to make them, we have to first do the process with mesenchymal stem cells. Dissect, centrifuge down to the pellet, put it in the flask, and then we add culturing medium, which is like the food for the stem cells. We put them in the incubators, which are like, think of it like a specialized oven where you can control the temperature, the CO2 levels, oxygen, all that. And they start replicating, but they're eating through that culturing medium in that time.